The next step in the recombinant DNA technology, the process involves the insertion of recombinant DNA which is present in the vector into the host cell. This is the next step. Now how it is done? Once the DNA has been ligated into the vector is made to get inside the host cell. As we had discussed how it is going inside the host cell, the cell is made competent using certain chemicals, and it is there are certain processes such as heat shocks which enables the cell to take up the recombinant DNA vector. Once it has been taken, then the next stage is identifying the recombinants and non-recombinants out of those cells. That also we had studied earlier. So it is a repetition of that thing only, and we are not going into detail. The recombinant DNA was what lied in the vector. The vector is to be transported into the host cell. When the host cells are taking up the vectors, they become transformants. Now using the selectable markers which could be involving antibiotic resistance, or it could be involving the insertional and activation technique where we have a chromogenic substrate that is used to identify the recombinants and non-recombinants, or the transformants and non-transformants, we identify which host cell is having the insert inside it, the recombinant DNA inside it. That means it has become a transformant. Once the transformant is obtained, the next step is obtaining the recombinant product, that is often a recombinant protein. So now we come to the end of this entire process because we are having our ultimate output. Now what is that ultimate output? It is the recombinant protein that will be generated inside the host cell with the alien gene which was incorporated into the vector. So the protein that would be generated by the recombinant DNA inside the host cell would form the recombinant protein. Now how do we obtain that recombinant protein? Certainly we would provide the host cell with the nutrient medium. They would grow in that nutrient medium. At the same time, the host cells which are having the vectors or the plasmids inside them, they are going to produce their copies. They are going to transcript that particular gene of interest or the recombinant DNA and form the protein. And what we have in the end as a result of these 5 stages in the process, we have a recombinant protein which could be packed and further in the various bioreactors and bioprocessors. It could be produced in a huge quantity, it could be packed and marketed. So this is all that you have to understand in the processes which are involved in obtaining a final product, that is the recombinant protein. How it is being produced? It is produced by recombinant DNA inside a host cell. Now for obtaining the recombinant protein from the host cell which has been formed by various stages that we have discussed in the previous lessons, you have to provide the host cells with nutrient medium. That could be provided in a small petri dish, or it could be provided in huge bioreactors. Now then once the protein is being produced, you can package it, and those rest of the processes after this are part of next lesson where we study downstream processing of this recombinant protein that has been produced. So we are going to take up bioreactors and bioprocessors in the next lesson, but for now we come to the end of the various processes which are involved in obtaining a recombinant protein, and that is what is the purpose of recombinant DNA technology.